Gonna get going here. And you should get out your clickers. It's the return of the clickers. Oh, really? Oh, I did say in the last class that we would be doing. What we're going to do for the next 3 classes is talk about fungi in a variety of different ways. And today, mostly, we're going to talk about the diversity of fungi, and then in the next few lectures, talk about some of their impacts and functions and functional diversity. In case you forgot, midterm is later today. Bring a pen, a number 2 pencil, and a photo ID. And I thought to reward everyone for coming here, we could do a few practice questions that relate more to the previous material than the fungal material. And if you brought your clickers, you can get, a half a bonus point or whatever it ends up adding up to per clicker usage. And I have now completely lost the... Well, maybe we won't use the clicker. All right, let's try it again here. All right, here we go. And then we're going to do them reasonably quickly here. These are the, kind of like some questions that may end up on the test, so I'll just give you until 45 seconds. All right, I'll let it go until a minute, and then I'm going to cut it off. So get in your answer. All right. The correct answer is C. They live only as, so this is kind of the type of thing you're expected to know about some of these groups. Not every single detail, but if we talked about some big feature, and you should be able to rule out certain other aspects of individual groups. So I'm just going to give you another example. All right, and this one I'm stopping at 45. Ready? Come on, come on. All right, so this may have been a bad choice of question. Did we not go over unicots this year? All right, everybody will get credit for A or C. Sorry. Unicots are a eukaryotic group that they ditched in the book this year for reasons that I'm not quite sure of. Blue-green algae is another term for cyanobacteria. But if it was the bottom 4 here, would you have been able to figure it out? Okay. The test won't have any. I just stole these from a quiz from last year. All right, I'm not going to do this as a clicker question, but let's just go through the next one here. Which of the following is not known to contain photoautotrophic taxa? And I gave you the answer, but pretend like you didn't see. I'm not doing it as a clicker. So you should be able to rule out some of these by process of elimination. You should know that cyanobacteria have photoautotrophs, diatoms and euglenids we talked about. We didn't talk in detail about all the stromatophiles per se, but we did talk about the apicomplexans all being intracellular parasites, so kind of awkward to be photoautotrophic when you're killing something from the inside. All right, and here's one last one. Again, not a clicker question. Which of the following groups is not known to contain multicellular organisms? Anyone have an idea? So C, there are some multicellular organisms in cellular slime molds. Apicomplexans, again, are these obligate intracellular parasites like Plasmodium that causes malaria. None of them form multicellular structures. All right, that's just, little bonus for coming in here today, and despite the stress of a test coming up. All right, so now we're going to get into fungi. There will be at least one clicker question in here. Depending on how far we get, there may be 2 or 3. So what we're going to do is basically go through a little bit about what are fungi, some discussion of their diversity of form, and phylogenetic diversity. I don't think we're going to get to the symbiosis part until the next lecture. So, well, there it is. It's right here. So a clicker question, sort of about fungi, but it doesn't require you to know what's in the next chapter, but it's based upon previous material. So I'm going to All right, until 1 minute. All right, that D, which 61% got, is the correct answer. So as you will see in a minute, actually I'll just go ahead to it now. The fungi are in this last one of the clades that we just briefly touched on but didn't really talk about in detail. That includes animals, choanoflagellates, and that clade is called the Epistacants. And the excavates are one of the other major clades of eukaryotes that they're not closely related to. So what we're going to do is focus in on this part of this clade today, the fungi. And if you look at the whole group, the Epistacants, you may not remember back to when we briefly touched on them, but they have a synapomorphy that they share in common, which is that if they have a flagellum, not every individual species, some of them have lost their flagella, but if they have one, it's present. There's a single one, and it's posterior, that is, it's in the back. And then fungi have a couple of synapomorphies that most of them have. That is, this is an event that happened on the branch leading up to the common ancestor of fungi. And the two key ones that we will talk about today and in the next few lectures are this thing called absorptive heterotrophy, which I briefly mentioned before and I will discuss again, and the presence of a particular compound called chitin, which is in their cell walls. And these are synapomorphies for the fungal clade. So later on in the course, we'll talk about the other members of this group, the animals and the choanoflagellates, but now we're going to focus on fungi. And I don't know if the volume's going to work on this. Is there any sound coming through? This is, I wouldn't worry about the words per se, the music's kind of nice, but it's this amazing time-lapse video of mushrooms growing.
Mushroom mycelium represents new growth, rejuvenation, regeneration. Fungi generate soil that gives life. The task that we face today is to understand the language of nature. My mission is to discover the language of nature of the fungal networks that communicate with the ecosystem. And I, in particular, believe nature is intelligent. The fact that we lack the language skills to communicate with nature does not impugn the concept that nature is intelligent. It speaks to our inadequacy of our skill set for communication. We have now learned that there are these languages that are occurring and communication between these organisms. If we don't get our act together and come in commonality and understand the organisms that sustain us today, not only will we destroy those organisms, but we will destroy ourselves. We need to have a paradigm shift in our consciousness.
That's not a fungus.
But I'm inadequate for the past to make a course change in the evolution of life on the planet. Okay, I tried. The fact is, I tried. How many people are not trying? If you knew that every breath you took could save hundreds of lives in the future, as you walk down this path of knowledge, wouldn't you run down that path of knowledge as fast as you could?
So this is a film made by this guy, Louis Schwartzberg, who actually was here, 6 months ago, giving a presentation at the Mondavi Center. It was pretty amazing. He has a TED Talk that has some other slow motion, incredible high-speed photography of like hummingbirds and bats doing pollination. Anyway, I'll post links to some of his other videos, but he's made this full-length feature movie about fungi with interviewing this one fungal expert that was just shown in the movie. And he has some great just shots of mushrooms in particular growing in particular environments. And then there's lots of diversity in the fungi. We will cover some of this diversity in this course. I just thought I would show you one of the coolest, sort of most unexpected things that a fungus can do. So this is a video of a fungal species that is predatory, and it captures nematodes, small worms, with a loop of its hyphy that we'll talk about in a minute, and then digests the worm.
Fungi are decomposers, but the soil-dwelling fungus, Arthropophis, is better classified as a predator.
So it's looped this worm and trapped it.
Just large enough to fit nematodes that live in the soil. If a nematode enters a ring, the ring constricts, trapping the nematode. Hyphy then invade and digest the nematode.
Anyway, it's just one of the examples. There's incredible morphological and functional diversity among the fungi, and what we're going to try and do in the next 2.5, 3 lectures is go through some of this diversity. So if we go back to those synapomorphies for the fungi, one of them is this key interesting aspect of their nutrition, which is helps define a lot of the ways that they function. And this is that fungi secrete digestive enzymes out of their cells. And those break down material on the outside of the organism. And then they have to bring into the cell the breakdown products. That's how they, or many of them, feed. And this is known as absorptive heterotrophy. It's heterotrophy because they're not fixing their own carbon. They're getting carbon from outside sources. And it's absorptive because they're absorbing this material after they digest it on the outside. And you will see when we talk about different types of fungi, many examples of how this sort of helps us understand their structure and function and their pathogenicity or the means by which they interact with other organisms. And this is an important term to understand for fungi in particular, this term called saprobes, which is that many fungi feed on dead material. So not like the pathogenic predatory one that I showed you the video of, but many of them will basically only effectively work on dead material or material that is otherwise sort of disassociated with the defense systems from some organism. There are plenty that are pathogenic too, and we'll talk about them, but a lot of them have this strategy, and that's really important to, for example, decomposition of biological material that, when a tree dies or leaves fall or other material falls, a lot of that decomposition is done by fungi.
In this sequence, saprobtic fungi and bacteria decay bread and fruit.
Sped
These
up.
organisms, which thrive on decaying matter, fill a vital role as the major decomposers in our ecosystem.
So you leave out some material, eventually fungi and bacteria will start to degrade it and then grow on it. This is just showing you black mold, which is one of the fungi. It's sped up. It's not. And basically, they really are. They're secreting lots of enzymes that soften up whatever material they're growing on, and then you have a lovely soup. And then they bring the material back inside via transport processes, but they're really unable to ingest in the same way that amoeba ingest, and they obviously not like animals in terms of their ingestion, so they have to have this absorptive process to get most of the material that they get from the world around them. So I'm just going to give you a brief introduction to the diversity of form first before we talk about phylogenetic diversity, when we really will talk about the diversity of form more. So when many fungal lineages, there are representatives in those lineages that spend their life primarily as single-celled organisms. Those are generally known as yeasts. And we'll talk a little bit more in detail about this one yeast, Saccharomyces Cerevisiae. That's, I think, what's referred to on my geeky microbiology shirt here. Saccharomyces Cerevisiae is what's used to produce bread and for fermentation for beer and wine, and we'll talk a little bit more about it later. But there are a diversity of single-celled organisms in the fungi that are called yeasts. It is not a taxonomic classification. So it's basically a description of a phenotype, and it is homoplasious. That is, it has evolved many separate times within the fungal lineage. Organisms that once were multicellular have gotten rid of their multicellular part of their lifestyle and become single-celled, free-living fungi. Most yeasts that have been characterized either live in liquid environments or very moist environments, or depend upon some sort of transition from dry to liquid environments to carry out most of their life cycle. They don't, as we'll see, the multicellular fungi form these structures called hyphy. The single-celled ones don't. They really just spend most of their life as single cells. And many different yeasts are really easy to work with in the laboratory, and so in addition to being used for producing fermented or other types of food products, They're also model organisms. So Saccharomyces Cerevisiae was the first eukaryote to have its complete genome sequenced. Genetic studies of that organism have revolutionized our understanding of how eukaryotic cells work, and we'll talk a little bit about that in the next couple of lectures. So the alternative to yeasts is basically, we're going to lump all multicellular fungi together, but as we go through the different groups, you'll see that they are not monolithic. They're not all the same in terms of their multicellular structures. But they have some simple sort of components that we can use to describe them all together. So this is a picture of a fungal, what's called a hyphy, which I'll show you in close-up detail in a second. It's basically the branching structure of the cells, and this is growing inside the xylem circulatory system of a plant. So you may be aware that fungi are pretty serious plant pathogens. In a lot of different ecosystems, plants can get infected with fungi. The fungi, as we will see, can get into their roots, can get into their cells, can get into their circulatory system. And if you have, imagine a fungus growing inside your blood vessels, that's basically going to create havoc for the plant that this is growing in. So multicellular fungi basically all are composed of a structure that's generally something like this. They're called hyphy, and they are these branched structures that give the sort of fungi, multicellular fungi, their characteristic branched formation. They are surrounded by cell walls. So each of these are individual cells, basically. They're surrounded by cell walls made up of this material chitin, which we will talk about in more detail in the next lecture. There are two different kinds of hyphy. Well, there are a few different kinds, but there are two primary different kinds that are defined by whether or not there are internal barriers between the nuclei. So in some of them, they have these things called septa, which are basically partial cell wall material, cell barrier material inside the hyphy. So when a cell divides, you get a nucleus up here and a nucleus down here. There's not a complete barrier between the two cells, but there's a partial barrier. And that allows for certain types of material to flow between the cells, organelles, nutrients, lots of other things. Even occasionally a whole nucleus can flow between these barriers. Whereas in these hyphae, there's another group of fungi that have no barrier at all. They are called Xenocidic, and you will see this referred to quite a bit in different parts of the book. So it's basically two major classes, and we will see how these affect the biology of different fungal lineages. And then just to point out that these hyphae, these branch structures, are really important for allowing for increased surface area to volume ratios. It's a general principle of biological life that you can create branched structures or folding over structures to increase the surface area to volume ratio. That's what happens in our brain. That's how the inside of our cells with all the different membrane-bound compartments work. And there are many lineages of organisms that use branched cellular structures to do this increased surface area to volume ratio. And one thing that we'll talk about a tiny bit, but the book talks about a little bit, is that this increased surface area to volume ratio is really helpful for absorbing nutrients. It's somewhat detrimental in terms of desiccation. So fungi are pretty prone to desiccation, and it's why you find a lot of fungi in higher relative abundance in environments that are moist. They don't do very well on average in environments that are very, very dry. There are a few lineages that have adapted to this, but most of them don't do well where it's not moist. So if you put together a whole organism based upon these hyphae, you can create structures based upon them, and there are two main structures. We will see a few other ones that we'll talk about a little bit. So there are Mushrooms, for example, if you look at this mushroom over here, they have underground structure that's generally referred to as the vegetative mycelia. Vegetative means it's not involved in sexual reproduction. It's just regular growth. And this allows them to spread out and search for nutrients, and also serves as an anchor for the organism. And then during sexual reproduction, many multicellular fungi will create these fruiting bodies that grow up out of the vegetative mycelia. And they're both made up out of hyphy. They're different in terms of some of their structures. They're different in terms of which parts of them have haploid cells versus diploid cells, for example, and we'll go over that. But in general, the multicellular fungi are made up of hyphae that are arranged in different ways, and it's the hyphae that together make up the body of a multicellular fungus. I just figured on test day we could have a lot of movies, right? So I'm not sure if this is of any relevance.
As the carnivore fungus expands, microscopic spores are released from gills on its lower surface.
So these gills grow on the underside, as we'll see in a little bit, of many of these mushrooms, and that's where spores come out.
As it matures, the nucleus of this young fungus furrows in on itself to better expose the spore-producing surfaces.
When you speed up their growth, they're just really cool. When you don't, they're a little slow and boring some of the time. All right, so not a clicker question, but I thought I would re-point out or rediscuss this issue of the branching structure. Again, it's a general adaptation to acquiring nutrients or to other factors that require increased surface area to volume ratio. So I've taken away all the taxonomic identifiers from this slide. Do you remember what these are? Anybody remember what they are? Yeah, so they're the Stromatopyles oomycete group. So I mentioned before that oomycetes have been frequently misclassified as fungi, and that's because they grow in these branch-like structures with compartments that look a little bit like hyphae. They're not the same. They don't have chitin, for example. They don't have some of the homologous structures inside, but from a gross outward, just examination point of view, they look a lot like fungi, which was why they were misclassified. How about these ones? Anyone remember? They are one of the slime molds. Anybody remember which one? Plasmodial slime molds also form structures that are a lot like the fruiting bodies of fungi. Many of them grow in these branch-like structures. I don't know if they were as misclassified as the oomycetes, but they also basically co-occur with a lot of fungi because they also prefer these moist, cool habitats. They are prone to desiccation as well. So you can see these growing, I think I mentioned this in lecture, after rainfalls in a variety of places on campus. Here's another one. Elaborate branch structures that look like hyphae. Yes, IgC gram-positives. Was there a question or? No, okay. All right, so I just wanted to point out that this has been a big issue in fungal biology, with organisms being lumped together into fungal lineages based upon appearance. And as we will see in a few minutes, it's also the case that there are multiple lineages that turn out to be fungi that don't have this branch-like structure for their hyphae in the same elaborate way. And they were misclassified out of fungi because of their lack of this fungal-like appearance. So it goes both ways. You can have organisms lumped together inappropriately or organisms split apart inappropriately. So before we talk about the different fungal groups, just wanted to give a little bit of preview of one of the main aspects of how the groups differ. So when people have compared the biology of the different major lineages of fungi, those lineages have now been determined largely by molecular sequence analysis, building phylogenetic trees, just like we did for bacteria, archaea, and the major eukaryotic groups. And now people have done the same thing within the fungi. They use a diversity of genes to do this, including sometimes ribosomal RNA genes, but in addition, many other genes. And when people have done this, they have found that one of the key differences or set of differences between fungal groups is in their life cycle in terms of how they reproduce, how or whether or not they undergo sexual reproduction, and how they produce the gametes that are going to be used for sexual reproduction if they have sexual reproduction. And so a general rule for most fungi, if you compare across them, is that the ones that are able to undergo sexual reproduction, that is frequently triggered by lack of food or some other challenge. If basically food runs short, many of them will go through sexual reproduction and produce what are called spores. We'll see these spores again in a minute. And then those spores, sort of the equivalent to seeds in plants or in the seed-bearing plants, but functionally very different. We'll see what they are. They can lay dormant in some environment for a very long period of time, in essence waiting for the conditions to improve before they germinate, before they grow and turn back into actual living, growing fungus. Many of the organisms use wind dispersal to transmit these spores. Not all of them, but many of them. So here's an example. These are called puffballs. They're a type of mushroom that when touched or when moved or sometimes just when triggered internally, they will release this massive puff of spores, billions of them frequently, to shoot up into the air to then be dispersed by wind. And I think, these are another type of dispersal of spores that you will see in a second. They're going to grow a little spore structure on the top of them, and then they shoot it out like a gun.
The black spore tip, a sac on top of a miniature water cannon that fires the spores beyond the ring of grass that has grown up around the cow pat and that the cow won't graze.
They have to get far away from the cow poo or they won't get onto living grass.
The spores that bombard the pasture are very sticky and will only be dislodged when the cow eats and digests the palatable grass. And there are other organisms.
And other organisms grab onto these spore bodies in order to transfer themselves around too. So they're so strong that you can get little worms climbing onto these little spore heads that hitch a ride, basically, for dispersal. And then this is a little video of the puffballs.
Produce spores within the fruiting body.
This is in real time.
The spores are dispersed by wind. When a drop of rain hits the puffball,
That's in slow motion.
Goes down through a small opening, giving them a boost for better dispersal.
So we'll go through some of what's on this slide as we go through the different lineages of fungi, but I'm going to go through it here just quickly. So all fungi are able to reproduce asexually. That is basically cells dividing to produce new cells. And we'll see when we go into the life cycles of different fungi that part of the life cycle of the fungi always includes some type of asexual reproduction. That is, cells making copies of themselves, generally by the process of mitosis. And that the cells that are making copies of themselves can be haploid or diploid, and we will see examples of both. There are some differences in terms of the exact mechanism by which different fungi undergo cell division. We'll talk about that later when we get into detail of the particular lineages. And then in terms of sexual reproduction, there are some lineages of fungi where no one has ever observed sexual reproduction. And we will come back to when I talk about the different groups, we will talk about them there. Fungi don't have anything that is by any standard definition male versus female parts, but they do have what are called mating types. So the cells or the adults appear phenotypically identical to each other, but they have a region of the genome, usually one gene, sometimes many genes, that varies within a population. And sometimes there are just two variants, so there's mating type A and mating type B. And in terms of mating, you have to mate with someone of the opposite mating type, like with male versus female reproduction. But again, there's no obvious distinction between the two types of cells or types of organisms other than this particular mating type. And we'll see a little bit more detail how they recognize a different mating type in order to trigger sexual reproduction. So we're going to be getting into, with fungi and plants and some animals, these life cycle diagrams. You should start getting used to them and studying them in more detail, because they're basically the critical way that we distinguish between the biology of many of these different groups. The life cycle of plants and animals will also be drawn in a diagram like this, with basically alteration between haploid phases and diploid phases in some of them, in others staying permanently haploid, in some of them spending most of the time as a diploid organism. And we see also some additional phases in the fungal life cycle. As we will see, there's something called the dikaryotic stage for a couple of the lineages of fungi. But basically, the way these diagrams work is we're going to start, you can basically pick anywhere on the diagram to start, and then the diagram shows the flow, basically, of cell types and multicellular individual types through time. And then certain types of processes, like fertilization, when you have haploid gametes merging together to create a diploid, is labeled. Meiosis is labeled. So meiosis is when you have a diploid individual producing haploid offspring, and then haploids can reproduce asexually by mitosis, for example. Diploids can also reproduce asexually. That's just growth of the diploid organism. And then on many of these diagrams, they might add an extra little few bells and whistles here. This is basically just showing what's going on with the different mating types, to make it clear that here's a mating type plus. It has to meet a mating type minus in order to undergo sexual reproduction. We'll go through, this is just the sort of generalized life cycle. We're going to go through these in more detail for each of the major types of fungi. So before we go into the biology of the different groups, I thought it would be useful to go through just a brief overview of what the different groups are. And then we'll come back to discussing their biology, their life cycles in particular. Well, we'll mention them actually throughout this. So phylogenetic studies of fungi have identified 6 major groups. This is how it's shown in the book, this phylogenetic tree of these 6 major groups. I just want to add here that it's actually unclear. Kytrids, this one lineage here, may be polyphyletic. There may be multiple separate branches that lead up to the organisms that are referred to as chytrids. And the same may be true for the Zygomycota. There may be multiple separate branches, so it's not clear if there are actually just 6 monophyletic groups or if Chytrids and Zygomycota might actually represent polyphyletic lineages. And I'll talk about that a little bit when I talk about the Chytrids. But we're going to go through them with the Microspheridia. Then we're going to talk about Chytrids, even though they might be polyphyletic, together, Zygomycota together, Glomeromycota together, and the most time we'll spend is going to be on these, the dikaryotic fungi, which are divided up into the Ascomycota and the Basidiomycota. And each of these have common names. I will probably switch back and forth between the scientific name and the common name occasionally. More likely, I'm going to spend most of the time, I will probably use the scientific name, but you should know both the scientific names of these groups and the common names. So you should know what's in this tree. We are now zooming in on one lineage of eukaryotes, the fungi. We're not, again, I didn't expect everybody to know everything about every single lineage of eukaryotes, but you really have to know at least these 6 groups, how they relate to each other, and a little bit of detail about the biology of each of these groups, as we will see. There's a table that will help you review some of the material that I will go through today. It's table 30.1 in the book. It has basically a summary of the features of each of these groups. It's not a lot of detail, but you should use that as your guide to know the lineages that are in the tree. And you should learn what's in this table too. And so I'm going to now, the last 10 minutes here, just quickly give you a brief tour, and then in the next class we'll talk in more detail about the biology of these groups. So the Microspheridia are one of the groups of fungi. They are now very clearly recognized as fungi, but for many years they were misclassified, not into the fungi, actually as protists that were particularly deep branching, and one of the reasons for that is they don't have a traditional mitochondrion. They have this organelle that is a little bit different than a mitochondrion. I think it's on one of these slides here. So they're called mitosomes. They're sort of like mitochondria, but not like mitochondria. And they were, in part, misclassified because they don't have these elaborate branching structures, and they don't have the standard mitochondria. But if you look at the Microspheridia in more detail, if you build phylogenetic trees of key molecular features like gene sequences, they clearly group with the fungi. And in addition, something that vexed people for many years that now makes sense, they have chitin in their cell walls. So the reason they have chitin was originally thought to have been due to convergent evolution. It is now thought to be synapomorphy for the fungal lineage. We don't have to worry about all the details here in this slide. This is just to give you a little bit of background on Microspheridia, and if you're interested in what they're like, there are some major major animal pathogens, including human pathogens, that are in this group. And we'll talk about them a little bit when we talk about pathogens later. As I mentioned before, this second, quote, group, the Chytrids, may in fact not be a monophyletic group. There's still some debate about this. We're going to talk about them together, but it is possible that some of the lineages inside Chytrids may actually represent additional deep branches within the fungal phylogenetic tree. Chytrids are most familiar to people as the causative agent of this major amphibian decline all across the globe. So there's a particular Chytrid that causes amphibian death, and it's been spreading all around the globe. There's a lot of work and a lot of research on this particular organism, including genetics and genomics and experimental studies. But there are other Chytrids that also cause a variety of problems that are becoming more and more studied. The Chytrids have a really interesting feature which distinguishes them from the other fungi. and that is that they are the only fungi for which the spores, so they produce these spores as part of their reproduction. And you saw in that video, those videos, that many of the fungi that people can, sort of look at, like mushrooms and these puffballs, they produce spores and launch them into the air in order to be dispersed by the air. Chytrids produce spores that can swim. So they have a flagellum, and they're able to swim through liquid environments, and this is unique in the fungi. Appears actually to be an ancestral trait that was then lost in the other fungal lineages. Again, you don't have to know all of the details on this slide. This is there if you're interested in learning more about chytrids. It has some of the features of them. Most of them are aquatic, that is, they live in the water. Many of them are parasitic. Some of them are just saprobic, that is, decaying, feeding on dead material. And anyway, there's a lot of detail here if you are interested in learning more about the chytrids. So one thing that the chytrids distinguish themselves from the other lineages is also in their life cycle. So for most of the groups, we're going to go through and compare them to each other in terms of how they go through their life cycle. And so if we look at the chytrids, you should be familiar with this from, not for chytrids, but in general for eukaryotes from previous classes. So eukaryotes can be diploid and then undergo meiosis. Diploid individuals can undergo meiosis to produce haploid cells. Those haploid cells then go through, in some cases, like with humans, very little else in their life. In the case of many plants and fungi, the haploid phase actually does a lot of interesting biology, grows into whole structures. We'll come back to that in a second. But at the end of that haploid phase, gametes are produced, and those gametes fuse together and via fertilization, and now you produce a diploid individual again. So this is altering between haploid phases of the life cycle and diploid phases of the life cycle. And all fungi, we can draw a diagram like this. In some cases, certain phases of the life cycle are incredibly short or, in essence, cut off. But most of them have a lot of interesting stuff going on in both the haploid and the diploid phase. So if we look at the chytrids, again, one of the things that distinguishes them when we look at their life cycle is that the haploid, you should study this life cycle. So the haploid spores, they're called zoospores. They're produced by this structure called a sporangium. So all haploid individuals here, those can swim. Again, in their aquatic lifestyle, this is very important to not rely upon wind dispersal, but to be able to swim. And if we go back over here, the gametes that are going to participate in fertilization also can swim. And so that's one of the key aspects of the life cycle that is distinct about chytrids. And you should look in more detail at this life cycle and the other life cycles in order to compare and contrast the different parts of them with each other. There's also this second group called the Zygomycota that also there's some suggestion may be polyphyletic. Again, we're going to talk about them all together here, even though it's still somewhat unresolved whether or not they're a single monophyletic group or not. Again, I've collected together information on this slide if you're interested about the Zygomycota. It includes that black mold that you saw in the video of the bread. Many of them are terrestrial. There's some very interesting biology in these groups. We'll come back to some of them when we talk more about their biology. And there's a diagram of their life cycle in the book that, again, you should study. There's also an animation that I'm going to show here, and I will post the animation to the website that goes through the whole life cycle of these, that'll help you understand the life cycle of these zygospore fungi. So I'll post this, and you can watch it whenever you want to. Good luck.